Now to Dave Cousins, who's going to chair the fourth session for us.
Just hi everybody, trying to get my video working. What a fantastic bunch of talks we've had so far. It's my pleasure to introduce the last session here. We've got a couple more talks, I believe. First up, we have Professor Imre Berger from Bristol. Imre is the Professor of Biochemistry and Chemistry there. He trained at Leibniz University, Hanover, and MIT in America. And he's had group leader positions at ETH in Switzerland and EMBL until joining Bristol in 2014. In 2017, he became the director of the Bristol Synthetic Biology Centre and the Bristol Biodesign Institute. And since 2019, he's been the founding and managing director of the Max Planck Bristol Centre for Mineral Biology. He's going to be talking about self-assembling thermostable adenovirus vaccine. And then in a last-minute substitution, Dr. Lucy Fairclough, who needs no introduction to this group, is going to talk to us about her work. But I will give Lucy an introduction. She's currently Associate Professor of Immunology at Nottingham. She trained in Hull and did her PhD at Nottingham Uni as well. And she has a long-standing research interest in allergic disease, asthma, T-cell biology, and also more recently in COPD. And she's going to tell us about cell-mediated immunity in COVID-19, the ACE cohort, and beyond. And I will hand over to Imre to start us off.
Okay. Thank you so much. So, I think I need very quickly, some advice how to start my, so do I start my Zoom or how do I do that?
If you just share your screen, at the bottom there should be a green arrow pointing upwards that says share screen. If you hold your mouse over your face.
Okay, let me see. It's like a free arrow. Sorry, I'm just, revealing my complete thing. Oh, share screen. Yeah, I have that. Okay, so just
and then
one moment. I thought that, that because I sent my talk to Alex, that it will be actually running automatically.
Oh, I see. I think it's easier.
Sorry,
to have control. Yeah.
Okay. No, that how to do it with share screen. That I should be able to.
If you get stuck, I can share the slides for you.
Oh,
but
I have it.
There you go.
Okay. Very nice. All right. So thank you so much for this really great possibility to join again after February. And so I showed the same slide, which I show here back then in the talk in February, but there I talked about the spike protein and the work we did on the spike protein, which you can see on the right. And today I will talk about something else, namely our efforts to generate a vaccine candidate, not only for COVID, we started this before COVID, but then of course when COVID came, then we also engaged there, and I will tell you a few things about that today. But I want to mention at this point that the work which I have introduced back then at the last Midland meeting, namely our project to bring this this antiviral or this drug, this small molecule which locked down the spike, as we had seen in our cranium structure, back then that has progressed very nicely. So we have now a product, actually, like an antiviral product, which we are pursuing with a spin-out Halo Therapeutics, which we have founded, and we are working towards MHRA approval for clinical trials there. But that's a topic then for another discussion. So I will talk today about this thermostable adenovirus vaccine. And this has been a story in my laboratory, which is going back a few years when we had been working on a component of adenovirus, namely this Penton protein. So on the left, you see the adenovirus, which you're all familiar with. So that's that virus which the Oxford people and the J&J are using to actually vaccinate against COVID. But what we were interested in was just one protein from the adenovirus, and that's the Penton protein, which sits there where these fibers are sticking out. And the reason why we are interested in that was because in that Penton protein was that because the Penton protein of some serotypes, not all of them, but some serotypes and some human and also animal serotypes of the adenovirus. They themselves, if they don't have the other components there, so the fiber and the hexon protein, they assemble in a virus-like particle, which you can see on the right, and that is this adomer particle, which my laboratory was working on. And adomer means adenovirus-derived dodecamer, because it is a dodecamer made out of 12 pentons. So you have 60 Penton Protomer molecules in there, which assemble spontaneously in the test tube in the absence of the other components of adenovirus into this dodecamer particle, which has incredibly exciting properties, which we actually don't really know yet why that is, but we are getting there. Okay, so what you can see here is a cryo-EM structure of this adomer, this adenovirus-derived dodecamer made of pentons. And because a movie says a lot more than many words, I will show you a movie. I hope that it will run okay, which tells you the background story. Because in 2019, what we were doing there, we were trying to make a vaccine against chikungunya. So there was a time, when chikungunya was an interesting virus and an interesting disease to work on. And this is a tropical disease which is slowly coming to the Western Hemisphere. And before COVID, that was considered to be a genuine problem. And we were at that time still in France when we started this work, and the French government was handing out money that one does something about developing a vaccine against that. So let me now pass over. I hope you will hear the words there, okay, to our own Adam Finn, who is a professor of pediatrics and the head of the vaccine center, and now the Pfizer Vaccine Center of Excellence here in Bristol. And you must have heard of him because he's now since COVID on every channel and in every newspaper.
Chikungunya is one of a group of viral infections that's spread around by mosquitoes. And it's just one of the whole bunch of these infections against which we have no treatments and no vaccines at present.
A major problem with vaccines at the moment is that they need to be refrigerated. In our latest study, we combined synthetic biology and cloud computing, two incredibly powerful techniques to create a new chikungunya vaccine candidate. We have developed a new class of synthetic vaccines. We call it the adomer. It consists of a protein which we have engineered.
This technology is unique because it doesn't rely on a cold chain. We can mass produce it at low cost, which is basically ticking all the boxes in what a very good vaccine should be.
Our ambition is to make vaccine candidates based on our technology against a whole range of human and also veterinary diseases. And we are very interested to see how powerful our technology really is.
Okay, and this was in October 2019. Okay, so there were a few prophetic words in there, and then when just 2 months later or 3 months later, this new virus, this SARS-CoV-2 virus, came out, then we of course had to do something about that, as I will come to at the end of my talk. But let me first explain a little bit why this adomer is so exciting and why it caught our interest. So it's a single component, so it's one protein from human adenovirus, and it spontaneously forms this super particle, this virus-like particle, which is the adomer. And it is exceptionally suited for multi-epitope peptide protein display. I will show you in a moment. Where you can deploy it to combat a wide range of diseases. It's self-assembling, so you just make this one protein component and then it forms automatically this very stable scaffold. It doesn't have any genetic material inside, so no DNA and no RNA. And at that time, so pre-COVID, this was considered to be actually something which is very positive. It is thermotolerant. It's actually thermostable, so up to 55 degrees. There's no cold chain required to store it, to produce it, and to transport it around. It's very, very well produced. I will show you this. Very highly soluble, so one can use it to give high doses. And it has a broad range of applications, and we are particularly interested in the vaccine side of this. And what you see on the left is a cryo-EM structure this endomer particle. So this was back then when we communicated this around 3 angstrom resolution. But as you know, electron microscopy has made incredible progress, and we have a structure now at roughly 2 angstrom resolution of this endomer particle. Okay, so on the left now we see the endomer in a little bit in a turned view. So we're looking right on one of these pentons there, which you can see in, so that's one of these 12 pentons, which you can see colored in different, in a rainbow color there. Each color is a different subunit. And when we now look at how well it can be produced, so in my laboratory, we have produced literally hundreds, if not, more proteins, also with this baculovirus expression system, which we developed. And I have never seen something expressing as well as this protein, this Penton Protomer, which then self-assembles into the decamer. So you can see this on the right, where we have the production of the endomer, which is so high that for all the laboratory work which we are doing, and even for preclinical work, we don't need more than a 50 milliliter culture, maximum of 100 milliliter culture to make substantial amounts of the dodecamer. And so the secret there is, and it's not a secret, but as you will see, is that you have loops on the surface which you can very easily functionalize, and there you can put in immunogenic epitopes, as I will show you, which we recapitulated in a library format in a synthetic gene, which we designed in a way that is very easy. And we established a 3-step procedure to purify this endomer, or like the vaccine candidates, which then have immunogenic epitopes in a GMP compatible setup. And we can, on the surface of one endomer particle, display 360 or even more epitopes. And they can be the same epitope, they can be different epitopes, they can be also T-cell epitopes from the same virus or from different viruses. And it's very easy to do because you manipulate it on the DNA level. And it's different than when you think about COVID, the COVID vaccines which are around, because we do not use the spike or the RBD there, but we use defined epitopes there. So it's like a focused epitope display technology. And here what we can see here is the Penton from the side, and that explains how it works. And so if you look at these Pentons from many adenoviruses, then you see that they have, there are 2 regions there which are not conserved, neither in sequence nor in length. They can be very short, they can be very long if you look through the species, and those are loop regions on the surface sticking out of the dodecamer when you have a dodecamer formation, and there you can put in these epitopes, whichever epitope. There seems to be no constraint evolutionary on the sequence content and on the size of these loops which are sticking out on the surface of the Penton and of the dodecamer. All right, and then came COVID. So what we did there was that we made, so basically before the structure even was known of the spike protein, when the sequences were out, then we looked at the sequences in early 2020, and by comparing to SARS-1 and comparing to MERS, we could decipher where the ACE2 receptor binding epitope was, and we grafted this epitope on the surface of the endomer particle. And within 5 weeks, we had the material ready and purified to do animal studies. And you can see on the right a negative stain cryo-EM of the endomer COVID-19 candidate, which contains these epitopes on the surface. This Achilles heel epitope, or the ACE2 receptor binding epitope, and it was ready for scale up within weeks. After we had actually gotten the sequence of the SARS-CoV-2 spike protein. And so I will now show you again a movie which will explain how we designed this endomer COVID-19 vaccine candidate. And this time I will have to provide the soundtrack myself, because this is just a movie. So you see here again a cryo-EM structure of this endomer, at this time at 2 angstrom, close to 2 angstrom resolution. You see here the Penton. This is the Penton Protomer, And what you see up there is the basis of those loops, so the base part of the loops, because you can't see the loops in the cranium, it's disordered. Here you see the receptor binding domain, and that is the ACE2, the binding motif, the RBM, so the receptor binding motif. And we lifted this off of the spike protein, and we fused it on the into these loops which are sticking out of the virus. Out of the endomer, and then when you express this protein recombinantly, then you assemble into this endomer particles spontaneously. But this time now you have the receptor binding motifs sticking out. And we have a startup in Bristol which prepared the Chikungunya vaccine candidate Immunology, and Immunology was the guys who then came to the laboratory during the lockdown. We did this work during the lockdown and prepared this COVID-19 vaccine candidate. And I would love to show you the immunological data and also like the in vitro immunization data, which we did down, and the mouse data there. But unfortunately, Morpheron put an embargo on this. But what I can tell you is that we had 3 routes of immunization: intranasal, subcutaneous, and intramuscular, and we got excellent neutralizing responses from there on the IgG level, and in the nasal mucosa also on the IgA level, which is particularly interesting because it tells you that this could be applied even without a doctor around. So that would make it really like a viable option for immunization in countries which will never get the Pfizer vaccine, and probably also not the AstraZeneca vaccine. And I think that with this, I come to the end. So these are the people behind this work. These are fantastic guys, and especially those, I don't want to single them out, but you know what happened back then when we started the lockdown? You could see that people, because with all of this uncertainty, then people were going home and wanted to stay home, and you couldn't blame them because nobody knew what was going on, and the university was shutting down. Only COVID research was still okay at the time, was allowed, and the people who did this work were volunteers who stayed because they wanted to do something about that. And this small group of volunteers did both the work on the spike protein, where we discovered then a potential antiviral, and also this work here on the endomer COVID-19 vaccine candidate. Adam Finn was instrumental for this because he put the Uncover group together, that is the Bristol University COVID emergency response group, where the animal people, so the people in the animal facility, were there the immunologists who helped us incredibly there. So we just had a retreat from the chemistry department where we had to describe our COVID experiences and our lockdown experiences. And you know, of course, many people suffered, many students suffered. So it was really a terrible time for many at the university. But when I was asked, I had to confess that scientifically, this time was maybe the most exciting time, because I was back again at the bench and working together with these dedicated people and trying to do something about the pandemic. So it was an incredible time. And with that, I would like to thank you for your attention and hand back to the chair of the session.
Many thanks, Imre, for a fascinating talk. We're going to do questions after both the talks, though, if that's okay, as we've done for the rest of the sessions. So it's now my pleasure to introduce Lucy Fairclough, who's going to tell us about cell-mediated immunity in COVID-19, the ACE cohort and beyond.
Okay, thank you very much. So my talk is I'm going to talk to you about 2 very distinct studies where we looked at cell-mediated immune responses after COVID-19 infection. So I'm going to start, and the vast majority of my talk will be on the ACE cohort, which is asymptomatic COVID-19 in education cohort, and I'll tell you a little bit about that on the next slide. But I'm also going to talk to you about another more recently funded study where we look at a patient group, a group of individuals that have vasculitis, and looking at their immune responses post-vaccination. The ACE cohort is very much a collaborative effort between colleagues at the University of Nottingham, but also with now very good friends of mine based at the University of Cambridge, Mark Wills, and I think he was on the call earlier, not sure if you're still here, Mark, and also Andrew Godkin from Cardiff University, who works with Bruce, who very kindly gave us a talk earlier. So this talk really should be in the cell-mediated immunity session, but because I'm stepping in at the end, because unfortunately we lost a speaker from the University of Birmingham, that's why I'm coming at the end of the day. So let me tell you about these two very different studies. So they're quite different in that in the first instance, that our research question with the asymptomatic COVID-19 education immunity cohort, which is funded by the National Core Studies immunity stream, was to try and understand immune response in natural infection with SARS-CoV-2 and any synergistic effect, sorry, let me just move that out of the way, of vaccination. And that's the second phase of the project that we've just got funding for in this ACE cohort. This is very much a collaborative effort between the University of Nottingham, University of Cambridge and Cardiff University, and very nicely supported by the National Core Studies. The other study that I'm going to very briefly talk to you about is a study in our individuals with vasculitis. And here we're not interested in natural infection, but we're interested in immune responses post-vaccination in these individuals that are probably going to generate very poor immune responses. So I'll start with the ACE cohort and the study design. So it was actually really fortunate that the University of Cambridge, Nottingham University and Cardiff University, nearly the start of the new academic term, as the pandemic was really raging across the UK, the three universities decided to set up an asymptomatic testing service, because we knew that young adults, as student populations, were likely to get infections but probably wouldn't have any symptoms. So it was very important to try and stop any outbreaks across the university sites and wider into the community, that we actually picked up these individuals that had asymptomatic COVID-19 infections. So what we did at that time was we offered these tests on a weekly basis to anyone that was on campus. So it was a PCR test done by the saliva sample. And at Nottingham, we also offered an antibody test from a finger prick blood sample. At the time of this asymptomatic testing, we also asked for consent to be contacted for future research. And that was really gave us a fantastic opportunity to enable us to recontact these individuals that had been via the asymptomatic testing service to come and enable us to do future research on these individuals. So we were lucky enough to get some National Core Studies funding where we could recruit a subset of these individuals into the ACE immunity cohort. So on the first visit, in the first phase of this project, we were collecting blood samples to enable us to isolate PBMCs so we could look at cell-mediated immune responses. But we also collected saliva samples and finger prick blood samples so we could look at antibody responses in asymptomatic infection and compare that to a group of individuals that also had symptomatic infection. At this point, I also, and this is very relevant to the conversation that was going on in the previous session, I also work with a psychoneuroimmunologist at the University of Nottingham. So we were also interested in trying to understand any psychological and behavioural factors that maybe were influencing immune response or even influencing whether an individual had caught SARS-CoV-2 infection. So at that first visit, the individuals also provided us with a questionnaire, including questions on things like, do you think you've had COVID? Have you had a positive test? But also well-being over the last few months. So that's phase one, and we finished that phase and we're just currently doing analysis of all of the data. And we're also very lucky in that we also got phase two funding from the National Core Studies, which will enable us to look at the influence of vaccination on immune responses. So here, and again, I'll show you the different breakdown of individuals that we've got in our study, but it'll enable us to answer all sorts of questions over the coming months. So what is the effect of vaccination in those individuals that haven't been infected? And how does that influence their cell-mediated immune response to vaccination compared to their antibody response? But also, what's the difference if an individual has had an infection previously, but that infection gave no symptoms, or that infection was symptomatic? And what is the difference in the cellular and antibody response post-vaccination? So we're just in the process of organising this second visit, and at the same time we'll be asking again the psychological and behavioural questionnaire. And then again, as part of this study, we'll be getting them back for a third visit, again to take antibody tests and blood samples and questionnaires. So in terms of where we got with phase 1, we were really lucky. Actually, in terms of the asymptomatic testing service, by the summer, we had about 35,000 individuals that had agreed to be contacted for any future research at these 3 university settings. In terms of what we were doing with the ACE immunity cohort and the specific samples we'd collected, again, by August, we've recruited about 550 individuals into our cohort. And across the 3 sites, we've got peripheral blood mononuclear cells stored in our liquid nitrogen stores. We've got blood spots and plasma and saliva samples to enable us to measure antibody responses. And during phase 1, we also worked really closely with each other across the 3 sites to harmonise the protocols that we were using to make sure that everything was the same, whether the sample was collected at the University of Cambridge or Cardiff University or the University of Nottingham. So like was mentioned earlier, we're really proud of the fact that we've got a fantastic cohort here to enable us to look at immune responses after SARS-CoV-2 infection or not, as the case may be. So in terms of the split of our cohort, we've actually got about 267 individuals which have, or those individuals do not think they've had a previous infection with SARS-CoV-2. Although, as you'll see from some of the data I'll present in a minute, it looks like actually some of those actually have had asymptomatic infection because they came up as antibody positive in our tests. We also caught a few individuals that were negative but had also been vaccinated. So unfortunately, that group doesn't give us a pre-vaccine response data, but it's still an interesting cohort for us to be able to generate some data from. And we've got virtually an equal number of individuals that have had a previous SARS-CoV-2 infection. And they've either had that, 161 of them have had that asymptomatically, so no symptoms at all. And 103 of them have had symptomatic infection with SARS-CoV-2. So we've got a fantastic cohort to enable us to answer some really important questions. 1 about basic immunity, but 2 about how previous infection will also influence antibody responses and cell-mediated responses to vaccination. So what have we been doing? Well, my main interest is cell-mediated immunity, and I'm a super keen person who uses flow cytometry, which is why I also chair the Midlands Innovation Flow Cytometry Group. And at Nottingham, we've got a spectral flow cytometer, the first only spectral flow cytometer in the UK that enables us to do deep phenotyping of T cells. And this is going to be really important in understanding T cell responses. So yes, we can do the AIM assay, the activator-induced marker assay, which gives us a fairly simplistic view of what's going on with the T cells. We can do floor spot assays, again, giving us a fairly simplistic readout in terms of whether you've got a T cell that recognises or has previously been exposed to SARS-CoV-2. But actually doing that deep phenotyping will be really important in understanding the quality of the immune response that's being generated. Again, both natural infection and post-vaccination. And as I've already said to you, we also collected some plasma samples, dry blood spots and saliva samples to enable us to look at antibody responses, so the humoral immune responses. And again, working very closely with a colleague at Nottingham, Paddy Ty, we've developed assays where we can look at the antibody responses to different strains of SARS-CoV-2, as well as looking at different classes of antibody responses and subclasses of antibody responses. And again, we also were interested not in whether you've just got antibodies, but how effective are those antibodies at neutralising virus. So I'm just going to really whiz past the data because I realise it's nearly the end of the day. So I think the key take-home message from this slide is that if you have got an antibody response against spike 1 of Wuhan, you're likely to also have a positive antibody response against different variants, although it doesn't always necessarily match, but most of those that have a positive response to one have a positive response to the other variants, which is really positive. We also looked at antibody responses split up in terms of our different groups. Again, I'm not going to spend ages because it's the end of the day and we're slightly over time already, but I think the key take-home message is, and I think this has been sort of mentioned already by Paul anyway, is the fact that if you have had infection and you've been vaccinated, you get much higher antibody responses, whether that's an asymptomatic infection or a symptomatic infection, you get much higher antibody responses and anti-spike responses post-vaccination compared to those individuals that just had natural infection or just vaccination, shown on the left-hand side here. So Cambridge and Cardiff are going to be doing the AIM assays and the floor spot assays, where we're adding overlapping peptide pools. These are commercially available overlapping peptide pools that we buy in, both to the spike protein, but also to membrane and nuclear capsid to look at any specific immune responses to SARS-CoV-2 infections, as opposed to any seasonal coronavirus infection. So the AIM assay looks at activation markers on CD4 and CD8 T cells, and the floor spot, as I said a moment ago, looks at cytokine responses. And what we're doing in Nottingham is, again, having isolated our PBMCs, Like Andrea, we freeze our cells down, and in discussion with Mark Wills at Cambridge University, we now have a method to get the cells back out of our liquid nitrogen store and keep good cell viability to enable us to do this deep phenotyping on the T cells. So we then culture our cells with these overlapping peptide pools, and then we stay in with a panel of 25 different antibodies, run it on the spectral flow cytometer, and that enables us to pick out populations of interest and do the analysis. So just a simple phenotypic identification where we pull out the lymphocytes. We're obviously only interested in singlets. We get rid of any dead cells that have been in culture. We have a dump channel where we get rid of any monocytes and B cells that we're not interested in looking at. We put CD56 into our panel to make sure that we can get rid of any CD3 positive, CD56 positive NKT cells, which are known to potentially contaminate T cell populations with flow analysis. And then we pull out those CD56 negative, CD3 positive cells to look at CD4 T cells or CD8 T cells. And then once we've pulled out those different populations, we can then look at things like cytokine responses shown on this slide. So things like interferon gamma and interleukin-2 for CD4 and CD8 cells shown across the top here. We can also look at those that are expressing two cytokines at the same time. So we can pull out our CD4 positive T cells, and then we can present interleukin-2 against interferon gamma and see how many of those cells are double positives. We also look at activation markers shown on the bottom right-hand side of this slide. But this is after a 6-hour culture, so you don't tend to get huge amounts of activation at this time point. But again, to look not just at T cell responses, but to look at the quality of the T cell memory response that's being generated is crucially important to understand both responses post-natural infection, but also post-vaccination. So again, in our assay, we add antibodies to enable us to look at different T cell memory populations. So things like CD8 TEMRA cells, naive cells, central memory cells. List goes on. You can split your effector memory cells into 4 populations by adding 2 further antibodies. So not only do we want to have a look at the sorts of cytokines that are produced, but we also want to know which cells those cytokines are coming from and what the T cell memory population is producing those cytokines. So we can put both of those things together, not just the cytokines, but also the cell markers that we use to enable us to look at what cells are producing what cytokines. So here again, we've picked out our populations of interest. And then we can look at activation and cytokine production of those cells. And in this slide, I must explain a bit more carefully. This is looking at cytokine positive CD8 T cells. And then we look within those cytokine positive CD8 T cells, what population of cells they're coming from. Is it a TEMRA cell? Is it an effector memory cell, etc.? So what this comprehensive flow cytometry enables us to do is to give us a deeper insight into the T cell response post-natural infection and post-vaccination. So we've done quite a lot in phase 1. So we've got the pre-vaccine samples, both from those that haven't had SARS-CoV-2 infections, those with asymptomatic and those with symptomatic infections. And we've collected all the samples, and we're slowly working through these 500 plus samples to try and generate some really important data. We've also, one of the other key aims was to optimize all of the techniques. So we've now got a comprehensive antibody screen and a panel of assays that we can run to look at antibody responses. And we've also optimized the deep phenotyping T cell response work by flow cytometry. I guess maybe a sort of slight sideline to mention is the fact that we have got different demographic groups in our populations of students. So this just basically represents data from the Nottingham cohort to show that although the vast majority are white, British, Irish or other, we do also have other populations that might be interesting in our research going forward. And as I said at the start, I also work with Kavita Vadera, so we're also interested in how mental health influences these immune responses, both natural infection and vaccination in these young adults. So just very briefly, I'm going to tell you about the Vasculitis Study that I'm also leading at Nottingham University. So here, this is a study to try and understand the quality of vaccination responses in those individuals with vasculitis. We're really lucky in that we've also got an age-matched control group that we can also look at antibody responses and cell-mediated responses post-vaccination. So this is just a quick slide to explain the study design for this part of my talk. So before the first dose, we took a blood sample, again, a blood sample so we could isolate peripheral blood and mononuclear cells, and those are currently sitting in my liquid nitrogen store. And we also took a finger prick blood sample so we could look at antibody levels pre-vaccination. Then individuals had their first dose, and 4 weeks after first dose, we took again another whole blood sample to isolate PBMCs and a finger prick sample to enable us to look at antibodies. Then we had second dose, and just before second dose, again, we had a quick blood sample taken, an antibody test done, and 4 weeks after second dose, we've collected some data again on antibodies and some blood to enable us to look at cell-mediated immune responses. And over the coming weeks and months, we're going to be collecting the 6, 9 and 12-month samples to enable us to try and understand the longevity of the immune response in these individuals compared to the healthy control age-matched cohort that we've also developed. So the end of my slides is really quick to say that basically we're going to be doing exactly the same things. We've got all the assays that are up and running. So from the finger prick sample of blood, we're going to be looking at cross-reactivity to different variants. We're going to be looking at antibody classes and subclasses, and we're going to be looking at viral neutralization. And of course, because I'm mainly interested in cell-mediated immune responses, we're also, having collected the blood samples and put them into our liquid nitrogen store, we're going to be stimulating them with these overlapping peptide pools to enable us to deep phenotype the immune response. And I think that will be really interesting in our vasculitis patients to see whether they've got a better or poorer T-cell response than our healthy controls. So like all research, this was done with a huge collaborative effort, all very last minute, to try and get samples in our biobank and to enable us to start this research. It was with a few key collaborators at the University of Nottingham, so Paddy that set up the antibody assays, Jonathan and Chris that set up the asymptomatic testing service at Nottingham, and Phil Quinlan that helps with data analysis. And also I put this picture up of my PhD students because they were all fantastic at working on the side of their PhDs and putting in the extra hours to ensure that we could make a fantastic biobank of samples before vaccination, which will enable us to answer those really crucial questions about pre-infection and vaccination. I also couldn't do this work without the two fantastic external academic collaborators that I work with, Mark in Cambridge and Andy at Cardiff. And of course, the flow facility at the university that support any of the flow work that we do. And again, a last quick mention to my NHS collaborators that again help get those samples. And last but not least, it always has to be the funders, and a massive thank you. So we were really lucky to get some National Core Studies funding to get this ACE cohort, this ACE immunity cohort off the ground and enable us to do this really important research in healthy young adults to try and understand what immune responses are like compared to some of these other cohorts that have been talked about today. And Vasculitis UK for funding our smaller study, trying to understand immune responses in individuals with vasculitis. So thank you very much.
Thank you, Lucy, for a fantastic talk. I'd like to open the floor for questions now. We've actually already got one for Imra, so I will start with that from the audience. It's from Lawrence Young. And he asks, what about T-cell responses to the spike when delivered by the adenovirus? And secondary question, will immune responses to Pentomer limit repeated vaccination with the adenovirus? And that was actually one of my questions as well.
Okay, so could you repeat the second question, because like that was too fast for me.
I'm so sorry.
So the first question, I understand that's about the T-cell epitopes. The second question is what again?
Yeah, will immune responses to Pentomer limit repeated vaccination with the adenovirus?
Okay, so those are both very good questions. So in terms of the T-cell response against the spike, we have concentrated on the antibody response to start with, because the neutralization occurs when you get like the binding against the RBM. But there is no reason not to also use T-cell epitopes, and we are working on that. And there are one advantage, or like one interesting feature of the adenovirus, is that it also has an integrin mediated, so it drains nicely to the lymph nodes, and it also has an integrin mediated, like an RGD motif there, with which it penetrates into the T cell. So it will actually deliver the T cell epitopes right there where you need them. So like we are working on this. It takes a bit longer, and we will find out about that. But I'm very hopeful that it will work. We didn't do this for chikungunya because we were told that in the chikungunya field, somehow people don't care about T-cell responses, so they only exclusively do antibody, or like, those who might be interested in picking this up, they are only interested in the antibody.
Yeah, maybe that needs to be revisited
Okay, so and then the second question.
Yeah, the second question was an immune response to the Penton fragment. Would that limit repeated vaccination?
Yes, so this is again a very good question. So, you know, like when we did test this by repeat injections in mice, but I mean, like so far we haven't seen like a detrimental issue there. But we have what we did is actually that we have looked for. We went through many serotypes of adenovirus, not only human serotypes, but also chimpanzee. Of course, you know, like, I mean, because that's so popular now, the chimpanzee adenovirus. So we went through that and also many others. And we found a whole series of Penton proteins, which make this dodecamer behave exactly like our adenovirus. And so basically what we think is that we will be able, even though that's not ideal in terms of like a production, from the production standpoint, but we will be able to vaccinate, let's say, against coronavirus with one type or one serotype, and then if it needs to be, then we can vaccinate against another infectious disease with, for example, the chimpanzee adenovirus. So both are excellent questions, and we are addressing them, and we have addressed them, and we have the tools to overcome problems if there should be.
Fantastic. So I had a couple of questions. How big an epitope can you put into the?
Very good question. You know, like, I mean, so let's put it this way: that we made bigger and bigger and bigger epitopes, and we were hoping that we would arrive at a point, you know, like, I mean, where the adenoma no longer forms, but we haven't reached the upper limit yet. So you can even put like folded protein in there. So you know, we basically stopped at. In principle, you know, like at like a small folded protein domains, because we had like nanobodies and these kind of things, because we didn't need to go further. But but it would be an interesting project for like a master student or something like this, just to, you know, like find the upper limit.
Yeah, the upper limit. Yeah. And my other question about the adenoma was in terms of scalability. How easy is it to mass manufacture?
It is incredibly easy to mass manufacture. As I said in my talk, that, you know, we had this facility, we have this facility also in my laboratory where when I was at EMBL, you know, like, I mean, we had many visitors coming and making proteins. So literally hundreds of proteins and protein complexes went through the lab. I stopped counting at some point, and many were very well expressed, but none of them were produced like this. So without any optimization, you get like gram amounts out of a liter culture. And, you know, right now we are optimizing it. So we have a GMP partner who is, you know, going through like establishing the protocols there. And it's probably one of the best produced protein, or the best produced protein I have come across. I don't know why that is, you know, like I'm trying to figure out why it is, but it's not so easy to actually find the reason why this is. Why is it so stable? Why is it so well produced? Yeah.
Well, it's good news, but it is. So, Lucy, I don't think there's any other questions from the floor at the moment, but I'm going. Oh, Andrea's camera's just popped on. Was that because you were going to ask a question?
I just wondered. So I think I must have. I was wondering about the questionnaire. I think that's what had prompted my thinking about it, because you mentioned it previously. And so I just wanted to follow up on that a little bit. What were the key questions that you thought would be most critical for the interface between the questionnaire and the immune response?
So I've done quite a lot of work with Kavita previously. So we know that sleep and stress has a huge influence on the immune response. So we actually took some questions from, I'm not sure if you've seen it, the Wellcome Trust COVID-19 questionnaire. So this is a questionnaire which was designed to try and ensure that everyone doing research and trying to understand health and well-being in COVID-19 used a similar set of questions. And then we also obviously asked questions about symptoms and experiences, etc. So yeah, it's a fairly substantive questionnaire that we've used, and I'd be happy to share it with you if you're interested.
Thank you.
Fantastic. So I had a couple of questions for you as well, Lucy. So you mentioned looking at the antibody responses to the different variants. Have you looked at Delta yet?
Delta is in the lab and waiting to be done, Dave. So I can't share any data with you today, but yes, we are going to be looking at Delta.
Cool. And I had another, so with the vasculitis study, you had a nice timeline laid out as to when you're going to be collecting samples, and I think you're going on for 12 months. Do you now need to factor in that the people are going to be getting a booster?
Yeah, so this is like everything COVID, Dave. It changes on an almost weekly, bi-weekly basis. So we had to make an amendment to our ethics to get a sample in. So we were due to get a sample in at 6 months, and then due to get another sample in at 9 months, but we've had to amend our ethics. So we will get a sample pre any booster and then 4 weeks post any booster. So yeah, it's an ever evolving situation with constant amendments to ethics applications to enable us to really answer the questions that come up, you know, as things develop and evolve.
And so my last question was around on your deep phenotyping slides, which looks very pretty, but you went through them quite quickly.
Oh, sorry.
No.
Conscious of time. Sorry.
Obviously, you were trying to show us the principle of the whole thing as well. But did I see that you seem to see a lot of CD8 positive naive phenotype?
Yes.
Cells.
Yes, but so the example I gave you, I can't remember what individual this was from. So I also did a study with NHS Coventry where we got some samples of individuals that had been hospitalised, and that data we're just in the process of writing up for a manuscript. So yeah, there's, you know, there are different populations depending on the cohort of individuals that you have a look at.
Yeah, it'd be fascinating to see it when it's written up.
Yeah.
Does anybody else have any other questions? In that case, I'll just like to say thank you to everybody for the presentations. I think, Lucy, are you going to do some closing remarks now?
Yes. Yeah, it's only just a few minutes at the end, really, just to say thank you so much to everyone for joining us this afternoon, and really a massive special thank you to all of our presenters who have shared their really exciting research. I think the one thing that for me has been fantastic this afternoon is how quickly we've had to evolve and try and develop our projects and work. You know, I think the terminology I heard used quite recently was stretch objectives, which basically means it needs to be done last week. And I think that's how probably pretty much every immunologist feels about COVID research. It's been a challenge, but it's been a fantastically exciting time for all of us to be involved in the work that we've been doing. So really, thank you so much to all of our presenters, and obviously also a massive thank you to the chairs of each of our sessions that have kept everything nicely to time for us all day. And also, finally, a big thanks to Alex, as I said at the start, who basically enables this session to actually work and function. And we can all be on a Zoom call and it all works beautifully for everybody. So thanks very much, everybody. And hopefully we'll have another session again. I'm not sure we'll do COVID. We might do some sort of other immunology session, but we'll keep you posted. And thanks very much for joining us.